Now, I would like to invite Surya Babu S to present a soil and water quality assessment in Pokali wetlands.
Okay, thank you, sir. Sir, am I audible?
Yes, you are.
Sir, can you see the screen? I have shared the slide.
Yes, we can see your slides.
Okay, good.
What is? Can you go to the presentation mode? Is
Okay,
it
okay,
possible?
thanks, sir. Good afternoon, all. My topic is integrated assessment of soil and water quality with microbial and plankton populations in Pokali wetlands of Ernakulam, Kerala. Let me begin with the introduction. What are wetlands? We know that wetlands are known as the kidneys of earth or nature
I
supermarket.
am just interrupting a little bit because your slides are very small. We cannot see it properly. If you can
Please go to the present mode.
presentation mode, yes, yes, then it will be visible to all.
Okay.
Yes, yes,
Okay.
yes.
It's perfect.
Okay. So we know that wetlands are the kidneys of earth or nature supermarket because of its ecological, wide ecological functions. And Kerala, especially our state, from India, it is rich with different types of inland as well as coastal wetlands, and also some protective areas such as Ramsar sites that includes the Coal, the Vembanad, as well as the Pokali wetlands. And the Pokali wetlands, here the sustainable agricultural method is practiced during the first 6 months, that is from June to October. Paddy cultivation is practiced, which is followed by prawn cultivation during the next 6 months. And I have analyzed the physical, chemical, and biological parameters of soil and water. And it is important to assess the pollution as well as the nutrient status and the ecosystem health. Then the microbes and planktons both are eco indicators. So by analyzing their changes, it is easily to interpret the changes in the pollution or the nutrient status changes as well as the ecosystem health. And the study, it helps to protect the environment as well as support some sustainable development goals. Coming to the aims and objectives, I have analyzed the physico-chemical properties of soil and water in 3 selected Pokali wetlands of Ernakulam district, analyzed the microbial as well as the plankton populations, correlated the abiotic and biotic parameters, and also analyze the interconnections between the soil, water, microbes, and plankton. And this provides a baseline study for the wetland conservation and agroecological management strategies. Coming to the materials and methods, 3 study areas were selected. From the slide, you can see that the first one is Valiyakadamakudi, second one is Erikara, and third one is Kotuvali. These three are Pokali wetlands. They are protected under the Ramsar Convention. Coming to the methodology, the soil parameters, that is a physical, chemical, and biological parameters of soil were analyzed. Some physical properties that were analyzed include the soil texture, color, and temperature. The chemical properties include the soil pH, electrical conductivity, and TDS. Soil carbonates, bicarbonates, chlorides, and calcium carbonates. Titration methods were used to analyze these parameters. Then the biological properties, that is a soil microbial characterization was done. Gram staining method was used and analyzed the microbes in the soil samples. Coming to the water parameters, the physical properties such as color, odor, and water temperature and depth were analyzed. Chemical properties such as water pH, electrical conductivity, TDS, chlorides, dissolved oxygen, and hardness were analyzed, as well as the planktons in the water column was also analyzed. Then the results and discussion. The soil parameters, that is a physical parameters such as the soil texture and color were analyzed. That is a soil is black soil with a sticky sandy texture. Then the soil pH were compared, and from the study, it was clear that the station 1 and 2 shows a slightly neutral or slightly neutral value, and the station 3 has a highly acidic pH value because the station was very much close to the in proximity with the Arabic Sea. Then the soil temperature of station 1 and 2 shows a medium soil temperature, medium value with a high value at station 3. Then the soil chlorides, 1.6 mg per liter of soil chloride were analyzed from station 2 and 3, and station 1 has a low soil chloride parameter. Then the TDS was very much high in station 2. Electrical conductivity as well as calcium carbonates were high in station 2. Then soil carbonates were similar value was noticed in case of soil carbonates as well as soil bicarbonates in station 1 and 2, with lowest value at station 3. That is the in station 3 both uh, that is soil carbonates and bicarbonates were absent. Coming to the soil microbes, station 1, 2, and 3 were analyzed. That is from the study. 4 colonies were isolated from Valiyakadamukudi area with 3 gram negative bacteria were identified from station 1, 7 gram negative bacteria from station 2, 2 gram negative bacteria from station 3. Then the water parameters, the color, odor, pH, temperature, dissolved oxygen, etc. were analyzed. The water was not much polluted, but it shows but it was not fit for drinking. Then water pH was slightly acidic at station 3. Then temperature was very high at station 3. Then the water depth was it varies from 6 to 5 meter depth. The fields were shallow. Dissolved oxygen was low at station 3. It shows some eutrophication in that station 3. Then TDS was very much high in station 3. Then station 1 and 2, the water chlorides was high. The electrical conductivity was high at station 2. These were the planktons identified from the 3 study areas: Serratium, Gonalex, Dinophyes, Gymnodium, all these species. Then the plankton abundance was moderate at station 1, with highest diversity at Edikara and lowest at Kotuvalli area. Then coming to the conclusion, that is, I have compared the water parameters and the soil parameters correlated with the soil microbial diversity as well as the plankton abundance in the 3 study areas. The soil temperature, electrical conductivity, and TDS was very high at station 3, and and also the soil pH was strong. It represents acidic condition, and these parameters contribute a stressful environment at station 3, and hence the microbial diversity was very low at station 3. And also the carbonates and bicarbonates were also absent at the station 3, hence these also contribute a stressful environment, and hence the lower microbial diversity was observed at station 3. Then coming to station 1, a moderate parameters, that is the temperature, electrical conductivity, and TDS was moderate in these 3 areas, and hence the microbial diversity was in moderate amount. Then the pH, coming to the pH value also, alkaline atmosphere was present in the station 1 and 2, and also, and then comparing the water parameters with the plankton abundance, we can see that station 2, it was favorable for the plankton growth, especially diatoms and some green algae was observed from the station 2, that is the Kotuvalli, Edikara area. Then the TDS represents the pollution and water clarity in the area, and hence the station 3, it shows a chemical stress, a high chemical stress, and hence the plankton richness and ecosystem plankton richness was low in that area, which represents reduced ecosystem health or low health of that ecosystem. Then dissolved oxygen, it was very much low. It shows a eutrophication condition in both in 3 study areas. Then electrical conductivity was moderate in station 3, which shows a moderate plankton abundance at the station 3, that is accompanied by limited acidic pH in that station 3. Coming to the summary, that is the Pokali wetlands, it comes under the Vembana Ramsar site. It is a nutrient-rich biodiverse ecosystem, comes under Central Asian flyway, and the integrated assessment of soil and water quality along with the microbial and plankton population shows that the study areas are dynamic and biologically productive ecosystem, and the high plankton diversity represents high primary producer community in that area, which supports which plays a critical role in supporting the wetland food web. And the physicochemical analysis of soil and water parameters, that is shows a that shows a spatial variability in these three areas, study areas. And correlation findings suggest that the water and soil quality, it has a direct influence on the biological productivity, especially the phytoplanktons, and also has an interdependence of biotic and abiotic components in the wetlands areas. And the study, it provides a baseline data for sustainable management and conservation efforts in face of climate change and urbanization pressures, and also contributes to some sustainable development goals. And these are some of the references, and thank you.
Thank you, madam. Thank you for this presentation. Now this is the time for questions.
I have a small question to ma'am. You have said that you have identified planktons, so you must have concentrated on phytoplanktons only, right?
Yes, ma'am. That is a It is a 1 year study, and I have analyzed only a few, out of which most were the phytoplanktons.
And you have shown that most of the areas have very low dissolved oxygen. So and station 3 is showing high abundance of plankton. So how it can be correlated with such small amount of oxygen? Planktons are surviving there. So it
Yeah,
is
I have already mentioned that there is no sole role of one parameter itself. So it might be it shows a temporal fluctuation, I think, coming to the dissolved oxygen, but it is supported by the TDS and soil carbonates. Some other parameters were favorable for the growth of such planktons in that area. So
so
dissolved oxygen, while ignoring.
is there any particular reason that the dissolved oxygen is so low in that particular area?
Actually, the study area, it is rich with fish diversity, prawns, and also they might. It is a prawn cultivation that season was very close to that uh, data collection time. So, due to the rich abundance of the fishes, prawns, and all those aquatic organisms, that may contribute to lack of dissolved oxygen in that area.
Okay, okay, okay.
Where these water bodies, aquaculture, or the water bodies?
Yeah, aquaculture as well as aquaculture is practiced during the 6 months. Paddy cultivation is practiced, followed by aquaculture. That is the fish, prawns, all those things.
So when fish cultivation and prawn cultivation is done, don't they aerate the water basically by using either fountains or basically other kinds of aerators?
No, sir, it's strictly natural. That is, during the 6 months, paddy cultivation is practiced, and after the paddy cultivation, they will clean up the paddy. That is a clean up the fields. It is close to the Vembanad Lake, so they will change the water with the coast. That is from the Bemrad Lake nearby, and then they are growing the fishes. That is, the fishes are cultivated in that area naturally. They are not providing any other aeration system or any other food materials. Also, that is, the fishes will grow with the paddy stubbles retained in the field. It is purely natural, and they are not aerating, or they are not doing any other agriculture practices in that area.
Okay.
Okay, sir.
Thank you, madam.
Okay.